Hello everyone. Let's begin with the next concept of this chapter 2, sexual reproduction in flowering plants. The name of the concept is outbreeding devices. See, now there are several plants which have inbreeding depression. Now how do they have inbreeding depression? That is continuous self-pollination that may lead to what? Inbreeding depression. So there are the flowering plants itself, they have given some devices to prevent the self-pollination and encourage what? Cross-pollination. Now what are these devices? The names of the devices, there are total 6 devices. The first one is dichogamy, the second one is heterostyly, the third one is self-incompatibility, the fourth one is unisexuality, diclany you can say, the next name for unisexuality, the fifth one is hercogamy, and the sixth one is the last one, that is diosy. Now, all these are the 6 different devices which discourage self-pollination and encourage what? Cross-pollination. So let's go into the detail of all these 6 devices properly. The first device which we already termed it as dichogamy. Now what is this condition? It is the condition where the pollen release and the receptivity of the stigma is not synchronized. Now to be more specific or to be more explanatory, I will say that is like the pollen, say for example the stigma, when stigma becomes a receptive organ, the pollen is not able to release, the anther is not able to release the pollen, or the pollen is not yet released, or vice versa. The pollen is released and the stigma is not a receptive organ. So this type of condition you term it as the device which is known as dichogamy. Okay. The second device that you can say is what? Heterostyly. Now heterostyly is what? In some species of the plant, the pollen and the flower and the stigma is placed at different position. So because of that, the pollen is not able to reach to the same flower. So what happens? There is a prevention of self-pollination. So the first two, that is the dichogamy as well as the second device which is known as the heterostyly. This prevent autogamy, which is self-pollination. The third one is what? Self-incompatibility. Now self-incompatibility means what? It is a genetic phenomenon where if the pollens are, it prevents the pollen to fertilize from the ovules. What does it do? It is a genetic phenomenon which prevents the pollen to fertilize to the ovules. How? By preventing it from what? Pollen germination or pollen to grow. The fourth one that you can say is what? Diclany or unisexuality. It effectively prevents self-pollination. It is the presence of unisexual flowers in plants that prevents autogamy but not geitonogamy. Here you can take an example of maize. The fifth one is what? Hercogamy. Hercogamy, it is seen in orchids where male or female sex organ themselves prove as a barrier to prevent self-pollination by some structural abnormalities. And the last one, that is diosy. In this, both autogamy and geitonogamy are prevented in several species like papaya, where male and female flowers are present on different plants, that is, each plant is either male or female. So the next process we have to learn about is about pollen-pistil interaction. Now what is a pollen-pistil interaction? The event from deposition of the pollen onto the stigma and till the entry of the pollen tube into the ovule is termed as pollen-pistil interaction. Now it is one of the dynamic process and it is an essential process for the fertilization of angiosperm to determine whether it is a compatible or incompatible pollen. Now to determine this, we have certain steps to be carried out. So the first step is what? Recognition of the pollen. Okay, for the pollen-pistil interaction, we need to know first step is what? Recognition of the pollen, that is compatible pollen, to be more specific. Recognition of the compatible pollen. The second one is growth of the pollen tube. The third one is entry of the pollen tube into the ovule. Okay, so the first one that we have to say about recognition of the compatible pollen. Now the pistil has an ability to recognize whether the pollen is compatible or right of the same species or and to reject the incompatible or the wrong of the same species or other species, right? So this particular capability, this particular ability of the pistil is given through the pollen itself, that is from the chemical components of the pollen that interacts continuously with that particular pistil, and that is the reason they can recognize whether it is compatible pollen or not. So after this process, after this thing is done, that is recognition is done of the compatible pollen, it further leads to what? Fertilization. Then we start with the second process, that is growth of the pollen tube. Here the pollen grains reach the receptive stigma of the carpel by the act of pollination. We already learned what is pollination in the initial go. Pollen grains, after getting attached to the stigma, absorb water and swell. After compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grain then move into this tube. Okay, so the pollen tube grows through the tissues of stigma and stylae, which is an elongated part of stigma, by secreting the enzyme that digests them. In some plants that shed pollen at two-celled condition, the generative cell, if you remember, divides and forms the two male gametes during the growth of pollen tube in the stigma. And in plants which shed pollens in the three-celled condition, pollen carries the two male gametes from the beginning. Okay. The third one, that is the entry of pollen tube into the ovule. After reaching the ovary, the pollen tube can enter the ovule either through the micropylar end or through chaliza, or you can also say through funicle or integument. Okay. It then enters one of the synergids through the filiform apparatus. Okay. Through the filiform apparatus. Then what happens? Where is filiform apparatus present? It is present in the micropylar end. The filiform apparatus directs the growth of pollen tube by secreting some chemical substances. All those events mentioned above together are referred to as what? Pollen-pistil interaction. All those events which when I say, that refers to what? It is about all the three stages. That is the first one, recognition of the compatible pollen. The second one, that is growth of the pollen tube. And the third one, that is entry of the pollen tube into the ovule, right? So all these events are known as what? Are referred to as pollen-pistil interaction. And the knowledge gained in this area helps what? The plant breeders in manipulating this interaction even in incompatible pollination to get desired hybrids. The next topic that we have to learn is about artificial hybridization. It is a crossing of different species to combine different characters or desirable characters to produce varieties. Okay, now this has been used by the plant breeders. It has been used by the plant breeders. Now why? In terms of crop improvement programs. Okay, now this is carried out by two processes. One is emasculation and second is bagging. Emasculation. Here what happens, the anther has been removed from the bud before it dehisces with the help of forceps. Now dehisces means what? Splits up. Okay, now what happens, the anther is removed from the bud with the help of the forceps. Now this happens in the bisexual flowers, the female parent which bears bisexual flowers. Okay, now second is what? Bagging. Now bagging is what? The emasculated flower is bagged or is covered with a bag which is made up of butter paper. Now why is it covered? So that there is no contamination to the stigma from other pollen grains to enter. Okay, now when the stigma of bagged flower becomes receptive, the collected pollen grains are dusted onto the stigma. The flower is rebagged and the fruits are allowed to develop. If the female parent is unisexual, emasculation is not necessary. Because we are doing it in where the female parent which bears bisexual flowers, right? So if the female parent is unisexual, emasculation is not necessary. In this case, what will be done? The female bud is directly bagged, and when the stigma turns receptive, suitable pollen grains are dusted onto it so as to allow germination and flowers are rebagged. The next process that we have to learn is about double fertilization. The next concept is double fertilization. The most important and unique characteristic feature of angiosperm is the participation of both male gametes in the act of fertilization. Now the pollen tube releases the two male gametes, right, into the cytoplasm of one of the synergids. Then the penetrated synergid starts degenerating. Now one male gamete fuses with the egg cell. Why? To form a diploid zygote, right? Now this process is known as what? Syngamy or generative fertilization, right? The diploid zygote which is there finally develops into an embryo. The second male gamete fuses with the two polar nuclei or the secondary nucleus that you can see. See, the first one which is there, it is getting fused with the first male gamete which is there gets fused with the egg cell to form what? The diploid zygote. The second which is there, what is it getting fused with? It is getting fused with the two polar nuclei or secondary nucleus in the central cell to form the triploid, that is primary endosperm nucleus, that is PEN, PEN that you say it in short. This process is called triple fusion, as three haploid nuclei are involved in the fusion. Okay, now after triple fusion, what happens? The central cell which is there, it becomes what? PEC, that is primary endosperm cell, which gives rise to the endosperm, while the endosperm develops into the embryo. As the fusion, syngamy and the triple fusion occurs, syngamy that is in the first male gamete, triple fusion that is in the second, right, occurs in an embryo sac, the phenomenon is termed as what? Double fertilization.